All right. I promise. Last video. So, just a few more things that we'll go through. We ended up with our idea of reproductive cloning, the idea that we can create an identical match to an organism. There is some concern about this. One of the things that we found with Dolly is that Dolly died a little young. Some people have suggested that even though she had old cells or old DNA. People often refer to the idea of the telomeres and everything that is why Dolly may have died a little young. There are some ways in which we can work around that, but again, this has not been hugely explored because there's not a lot of possibilities for this. Now, there is another idea out there called therapeutic cloning, which is the idea of instead of cloning an entire organism, cloning a part. For example, I have a damaged liver for some reason, and we could grow me a new liver and then take out my old used liver and put in a new liver. Um, that is an idea that is very much in the works and very much progressing towards practical uses. I mean, there are definitely have been situations where they've done it in certain cases. Skin grafting is probably the most common example of that, and they're getting better and better at that. There's a cloned cat. This is the idea of that therapeutic cloning, much more practical. We take stem cells and create new types of cells. It's just a matter of finding the right cells. Um, the cells that we find that work the best are things like bone marrow, mammary cells. Those seem to not have a huge memory for what they were, so they can turn into other cells. That being said, the best are embryonic stem cells. And one of the controversies surrounding that is an embryonic stem cell, in order to harvest an embryonic stem cell, you have to destroy the embryo. Obviously, that's not the long term goal because what we're really trying to do is be able to figure out how to get stem cells from adults to replace things that are going wrong in that organism. Um, this is the idea of an RNA type vaccine. We'll skip that. Okay, you may have heard about the idea of CRISPR. This is became popular in the news. It's a technique that has grown in popularity. It steals some techniques from bacteria and actually allows you to sort of inject everything you need at the same time into a cell. It cuts some DNA, it inserts a new gene, and it allows you to put a new gene into place. Where does this leave us? Well, there is a unsubstantiated case where this was used on humans. It was not supposed to happen, but there's a doctor that claims he did it, not in the United States, and there's still a lot of controversy surrounding it. Doesn't make a lot of sense what he did. He actually took a gene that gives people HIV resistance, which is great, but not something that it's preventable in other ways. You can avoid getting HIV in lots of other ways. You don't need to insert a gene. It seems like not a very practical first case for using CRISPR techniques in humans. But that's one of the cases out there, and you can look up more information on that. So, moving forward, where do we go next? So, there's a whole thing out there, and this is a short list that is not complete, but the idea of using DNA technology in forensics has been around for a while, and we have lots of sitcoms now, CSI, NCIS, all these shows out there, these crime shows where they use these techniques. One of the ones that is they don't talk about in terms very much, but it's the VMTR. This is the standard in DNA fingerprinting these days. And it's really kind of an interesting thing because in the old days, they actually needed two samples of DNA and they need to run them side by side on a gel. You don't need to do that anymore. What they're looking at are these areas of your DNA that are highly variable and do nothing. So they what we do is we look at how many times these small sequences are repeated. So there might be a, GAATC repetition, and in some people, it might occur 20 times, and other people it might occur 24 times. And what it does is it looks at your chromosomes and sees how many repeats you have and just records that number. So what it ends up being is a record of that is just a number. So if you have a DNA fingerprint in a database somewhere, which my guess is most of you don't because you haven't committed a felony yet, hopefully. But that's how you get your DNA in the system. The other ways to get your DNA in the system right now are to be in active duty in the military and also to be a police officer, especially ones that investigate crime scenes, because they want to make sure they have those people cataloged so that when they pop up at a crime scene, we know that it's possible that it was just a contamination of somebody that was at the crime scene. And we're not looking for somebody that, is right in front of us, in fact. But yes, these techniques are pretty cool. And the best part about it is that we can use this technique for cold cases too. Cases where we do not have DNA for many, many generations. There's the famous case with in California, the Golden State Killer. What they did is they actually found some DNA that was from the crime scene, and they sent it off to a lab pretending it was John Doe and said, "Hey, this is, they did not know who it was from, but they knew it was the perpetrator. They assumed it was the perpetrator, and they sent it off to these, I don't know, I think it was 23andMe, and said, "Hey, my name's John Doe, and I want to know if I have any relatives out there." And they came back with a report that said, "Oh, here's a couple relatives that are in our system." And of course, then they track down the family tree of those people and find a guy that is likely to have committed the crime. And then they chase after him. They find a coffee cup that he leaves and steal the DNA from it. And then they have his DNA, match it, and they arrest him. 20-year-old cold case. And it's a good thing he's in jail. He committed some horrible crimes. Other ideas, we can clean up environmental toxins using this. This is a really cool idea. Agriculture has been huge. There's a company called Monsanto, which if you haven't heard of it, they're huge into producing these agricultural uses, pesticide resistant, herbicide resistant. So they make a lot of crops that are resistant to the chemical that is in Roundup. And so what you can do is you can wait till your plants are sprouted, but along with that are going to come weeds, and you can spray your whole field with Roundup essentially, and it will kill all the weeds, but won't kill the plants that you want. They're trying to put nitrogen fixation into plants like corn so that they have to rotate crops less, soybeans and things like that. Frost resistance, so, get ready for orange trees growing in Pennsylvania. That's a possibility out there. Growth hormones, getting things to grow faster, high yields. Nutrition and taste, these are things that are only starting to be looked at because this isn't the way that the agricultural industry works. People don't look at a tomato and say, is it nutritious? They look at it and say, does it look like a good tomato? And that's the most important thing. Does it last on a shelf? Can you get it to the store? Can it look good? Will people buy it? But they are starting to. They actually added some nutrition to certain rice strains. It's not popular in the US, but it is worldwide. They even put some animal vaccines in there. And then there's so many pharmaceutical ideas, gene therapy. There is cases now that they're using gene therapy, designer babies. That's sort of a pie in the sky idea, but I wouldn't be surprised if we start seeing them producing bacteria or protein supplements, DNA vaccines. We have cavity fighting bacteria. That's another one. Artificial antigens. So that's one that is now really actually being produced. The big one out there is these things called CAR T cells. CAR T cells are simply what you're doing is you're taking your white blood cells, shipping them off to a lab, and they actually insert a gene into your white blood cells that makes it produce a specific protein antibody that is designed to fight a specific disease. Then they send the cells back to you, and you inject the cells back into you, where they grow and behave like normal T cells, and they are fighting off whatever you are looking for these to fight off. And there have been some really successful cases with CAR T cells. It's a really promising idea, and they definitely have some good backing to show that this works. However, it's incredibly expensive at this point. We're talking over $1 million per case. So it is something that is not quite at the level of getting a little antibiotics at the store. So it's more to come on that, but the price should drop, and hopefully we'll see that for some pretty tough to cure diseases out there. So, lots of exciting things to come. All right, thanks guys for paying attention. Bye.